bugs big bombs. Please welcome Mr. Bill Gates.
We also face a new threat that the next epidemic has a good chance of originating on a computer screen of a terrorist intent on using genetic engineering to create a synthetic version of the smallpox virus or a contagious and highly deadly strain of flu. So the point is that we ignore the strong link between health security and international security at our peril. Whether it occurs by the quirk of nature or at the hand of a terrorist, epidemiologists show through their models that a respiratory spread pathogen would kill more than 30 million people in less than a year. And there is a reasonable probability of that taking place.
The choice of H5N1 for our research, we really tried to hit two birds with one stone. On one hand, we tried to address the very fundamental question of how influenza viruses become airborne. By using H5N1 as the model virus, we would directly assess the risk of H5N1 becoming airborne. Particular genetic changes that have occurred during the pandemics of 1918, 57, and 1968. And we used that knowledge to genetically modify H5N1 bird flu virus and try to see if it would also acquire this ability to become airborne. And initially that was not the case. But we then did an experiment of natural selection in the ferret model by serially passaging the mutant virus in ferrets, in the nose of ferrets. And after 10 passages in ferrets, the virus had become airborne, so it could be transmitted by coughing and sneezing and breathing between ferrets. We also characterized the viruses that were transmitted by the aerosol route to identify the mutations that were associated with airborne spread. And that it was actually fairly simple for that virus to acquire that ability. It only took in between 5 and 10 substitutions in a few genes to become airborne. Genetic changes that occurred in the virus were quite similar with previous pandemic viruses. And we further investigated whether this virus would be sensitive to antiviral drugs. Exactly which mutations or which biological traits we have to be on the lookout for during outbreaks, we might be able to eradicate those viruses specifically that are a direct threat to public and animal health. We can better assess the effectiveness of antiviral drugs and vaccines before the viruses actually emerge in the field. Security issues around this manuscript are really minimal. We perform our work in a very safe laboratory. The facility is highly secured also, and is under inspection by two governments, the Dutch government and the US government. You need very trained specialist virologists to do this type of work, and so it's unlikely that any lone wolf would repeat this type of work. On top of that, we show in this manuscript that the virus is not particularly lethal once it's transmitted via the aerosol route. In ferret, none of the ferrets die, and it's not particularly efficient in transmission, so it would be unlikely that if this virus would be released from the laboratory, it would actually cause a massive outbreak. The future of this research is really to increase our understanding about the specific mutations that we identified, the biological traits that are associated with those mutations, so that in the future we really can predict which viruses in the field are more likely to become airborne at any given point in time. Increasing our fundamental understanding of the genetic changes that are occurring in viruses once they become airborne. And also research on how generic our findings are to other influenza viruses. But these are really fundamental projects for which much more research work is needed.
The opening of China's first laboratory for level 4 pathogens, or P4, in Wuhan, central China's Hubei province, recently is seen as a great achievement for the country. P4 refers to microbiology laboratory safety procedures to handle level 4 pathogens, that is, the most virulent viruses that have a high risk of person-to-person infection, of which Ebola is one. Biosecurity has for long been a worldwide concern. For instance, 2 lab researchers working on the SARS virus at the Center for Disease Control and Prevention in Beijing were infected by the deadly virus after it was accidentally leaked because of some employees' negligence in 2004. The infections spread to a few more people before being arrested, and 5 top officials of the center, including then-Director Li Liming, resigned after being held accountable for the accident. Worse, in 2011, 2 scientists, Yoshihiro Kawaoka of the University of Wisconsin-Madison and Ron Faucier of Erasmus University in the Netherlands, succeeded in tweaking the already dangerous H5N1 bird flu virus in the lab into a more easily transmittable pathogen among humans. That created an uproar in the global science community and raised public fear that terrorists could use the example to create a lethal biological weapon. Scientists investigated a virus called SHCO14, which is found in horseshoe bats in China. The researchers created a chimeric virus, made up of a surface protein of SHCO14 and a backbone of a SARS virus that had been adapted to grow in mice and to mimic human disease. The chimera infected human airway cells, proving that the surface protein of SHCO14 has the necessary structure to bind to a key receptor on the cells and to infect them. It also caused disease in mice, but did not kill them. Although almost all coronaviruses isolated from bats have not been able to bind to the key human receptor, SHCO14 is not the first that can do so. In 2013, researchers reported this ability for the first time in a different coronavirus isolated from the same bat population too. The findings reinforce suspicions that bat coronavirus is capable of directly infecting humans, rather than first needing to evolve in an intermediate animal host, may be more common than previously thought, the researchers say. But other virologists question whether the information gleaned from the experiment justifies the potential risk. Although the extent of any risk is difficult to assess, Simon Wayne Hobson, a virologist at the Pasteur Institute in Paris, points out that the researchers have created a novel virus that grows remarkably well in human cells. If the virus escaped, nobody could predict the trajectory, he says.